Well, welcome Cancer Patient Lab Meeting Access, the latest developments in liquid biopsies. We have Lauren Leiman and Jennifer Dickey here today. Lauren is the Executive Director of BloodPAC, that's the Blood Profiling Atlas in Cancer, and Jennifer Dickey is of a subsidiary of LabCorp, and they're going to lead a discussion on the cutting edge of liquid biopsies. And with that, I'm going to turn it over to you, Lauren and Jennifer.
Thank you guys for having us. It's lovely to meet you. And BloodPAC is a very relatively informal organization, so we encourage you to ask us any questions. Feel free to interrupt us so you don't lose your train of thought. We're very easygoing. I also want to say, in addition to me, we also have my colleague Cheyenne Jankowicz, Donna Elbers, and Doris Lopez, who's here. I'm looking for everyone on the screen from BloodPAC. So Cheyenne manages all of our programs. Doris is our head of communications, and Donna is our head of data. So if there are any specific questions that you have around any of those topics, you can feel free to ask them as well. A brief introduction. I'm Lauren Leiman. I'm the executive director of BloodPAC. About 8 years ago, I was at the White House as the head of external partnerships for the initial White House Cancer Moonshot. And Peter Kuhn, who is with us here today, came into our office to talk about liquid biopsy and why it was really something we should be tackling and how we could tackle it through a commitment to the White House Cancer Moonshot. About 10 months later, we had established a mission statement, which was to accelerate the development, the approval, and ultimately we've added the accessibility of liquid biopsy technology for cancer patient benefit. We had decided we were going to do that by creating frameworks and standards for the community at large to use, to also aggregate data to support those standards and frameworks. And then we also have felt again over the past few years that we can be successful in those first two areas, but if we can't get this technology into patients' hands, what is the purpose of all of our hard work? And so we have created kind of an accessibility, stakeholder engagement workflow as well that focuses on everything from regulatory issues, reimbursement issues, accessibility and education issues, all of those things involved. And so Peter, I'm excited to say, since he's here, is a co-founded OG BloodPAC member, co-founder, and sits on our executive committee and our scientific co-chair committee, and helps guide the organization. Jenn Dickey also sits on our scientific co-chair committee and is an original member of BloodPAC as well. So I can share my slides very briefly because I'm not going to spend, I want you guys to ask us questions. Let me see if I can do this. Can you guys see this? Yeah, okay, great.
Yes.
And I'm just going to show briefly what it is. I know it's not in like slide mode, but because I don't know how to do it from Google. But we were formed, again, as I mentioned, during the White House Cancer Moonshot, first one back in 2016. In 2017, you should know, we are a nonprofit. We started with an original group of about 20 organizations, pharmaceutical companies, diagnostic partners, academics, foundations funding in the space, and in several government agencies to help, again, achieve our goal of accelerating the development, the approval, and the accessibility of liquid biopsy for cancer patient benefit. As I mentioned, we do that in 3 different areas, 3 large buckets. Kind of evidence generation. I'm not sure if I love that title anymore, but it's really aligning around frameworks, creating standards for the community to utilize, aggregating the data to support those, or running, in some cases, we do run a few different prospective studies to generate that evidence as well. And then we have this larger stakeholder engagement bucket. Today, we have over 74 different organizations that collaborate and are members of BloodPAC. Again, the only addition to the original that I mentioned from the original 20, the diversity from the first 20, was we have started to add private payers to the conversation recently, which is, I think, an added benefit to what we're capable of doing and also shows the progress of the field generally. I think this gives you a good idea of any kind of visualization of who's involved in BloodPAC, so you can see the depth and breadth of who we work with. I will stress that, again, we are pretty heavy on the diagnostic and pharmaceutical side, so the corporate side, and have very few academics who work with us, primarily those who have been kind of leaders in the field for decades, who have been pioneering this space and have the capacity to kind of help guide us as a whole. We also, just so you know, work again in those 3 buckets. We work in working groups. Jenn also leads several of our working groups. As Peter said earlier, she's the head of our regulatory working group. She also partners with us on our blood collection kit project that falls underneath accessibility. So with that, though, I kind of, now that you know a little bit about BloodPAC, I was going to, I'll stop sharing my screen for a moment. And Jenn, if you're give you a second to come off mute. I think we should start at the beginning if you're okay with it. And I'm going to ask you maybe to define liquid biopsy. What is it? What are we, and then also maybe talk about what are the 3 primary uses today and the future for liquid biopsy. I know for many of you, this may sound extremely kind of beginners, liquid biopsy 101. However, we have submitted actually articles to STAT and we've had them accepted. And it seems that there's confusion today still around what we can do today, what the uses are, the potential, what we can do today, what we can do tomorrow, and what we can potentially do tomorrow, and where we're aiming to take the industry as a whole. So I think that might be a nice general background to give everyone if you're able to do that.
Oh, absolutely. Well, thanks for passing it off to me, Lauren, and giving me the hard task of defining like a very broad field of study, really. But in essence, if you think of liquid biopsy, it's the notion that a biopsy traditionally would be where one takes tissue from the body to examine it clinically to understand its characteristics. Many, many decades ago, it was discovered that tumor cells and tumor DNA float in the circulation of the body when they're present. And so a liquid biopsy is basically using biofluids, generally blood, but taking that circulating tumor DNA or cells out of the body and doing clinical examinations on that tumor DNA that's available in your blood. And the reason this is a powerful tool and one that we advocate strongly for in the oncology community, and one that we actively try and develop and promote the development of new assays for, is because it is less invasive than a standard biopsy and has a lot of functionality that can help improve patient lives. So kind of breaking it down into the 3 buckets of things you could think about immediately using liquid biopsy for. One is similar to a biopsy. Take blood from a patient and examine those tumor cells or tumor DNA for the characteristics that would help a clinician determine a therapeutic odyssey for that patient, right? Help them find the variants and genes that are mutated that would guide them towards a precision medicine solution. And this is great for liquid biopsy because there are frequently patients with cancer who are too medically fragile to undergo a full biopsy. And so this gives a quicker and easier, less invasive way to obtain that information, particularly for patients who aren't able to provide a biopsy. The second obvious use, and one that gets used already today, is for monitoring. So as a patient's therapeutic journey progresses, they may develop resistance mutations or DNA signatures that would indicate when it's time to change a drug or reconsider a therapeutic option. And that monitoring, while you can re-biopsy a solid tumor, monitoring in a low invasive way with blood is actually an extremely powerful way to make sure that the therapy is there and it's working. Now, moving into the tomorrow, moving into the next step, we can take that monitoring and then use that gain sensitivity of our assay, gain a sense for understanding when we have what we call molecular residual disease detection. So this would be like prognostically saying whether a therapy is working and therefore they don't need any additional therapeutic interventions, or maybe that there may be an early sign of a relapse or recurrence of the disease. And so maybe we could get to treatment earlier, get ahead of progression and move it towards a happy therapeutic outcome quicker. The other kind of bucket, of course, is then moving earlier in the cancer continuum. So this would be what's called multi-cancer early detection or detection. So this would be the notion in the future where we're going is the ability to assess apparently healthy individuals, people who don't have a diagnosis of cancer, and then determine early or in time for meaningful clinical interventions that they have cancer, and in this way prevent the growth and development of advanced cancer if at all possible. So those are kind of the buckets, as Lauren likes to call them buckets, the buckets of kind of analytical studies we look at. We look at things for comprehensive genomic profiling for treatment. We look at things for molecular residual disease for prognosis or monitoring. And then we look at multi-cancer early detection or detection to help as a screening tool to detect cancer better and earlier. And all of these with a number of different technologies, though many of them involve next generation sequencing technologies and other kind of more advanced diagnostic techniques, which makes it an exciting adventure. And where I come in is then an exciting discussion with the FDA about how those products and services should be regulated and make sure we're all on the same page.
Thank you, Jen. Can I ask you to tag on one thing? So as someone working for a company, PGDX originally, that was developing these tools, why is it so hard and why do so many of them not succeed? And I think is a really a level ground setting kind of something we should set, establish kind of a basis for, so people understand how hard this is and why we've been working for 8 years now and I don't have a new job.
Yep, Lauren is not in danger of losing her job anytime soon. I'd say it does actually take a lot of time, energy, and investment to bring new cutting-edge technologies to the clinic. And part of that is because of the time and expense of clinical trials. And another part of that is as the clinical field evolves, it takes time to align to analytical standards, clinical standards, and acceptable, even the lexicon of how we are going to describe these clinical scenarios, how we're going to define what is good enough, how do we define what's a clinical response. Getting to these things is very difficult. I was previously at the FDA, but I left the agency and went to industry originally with a small startup company, Personal Genome Diagnostics. We have since been acquired by LabCorp Oncology. But one of the reasons small companies struggle is the technology does not start off being cheap. It needs to be developed, which takes an extensive R&D development operation. And then getting it through a regulated body, getting it through the FDA, getting it through reimbursement, getting it through all of these things requires the generation of evidence. The generation of evidence is generally clinical trials, prospective, retrospective, analytical studies, comparative studies, et cetera. And these studies are expensive, difficult to design, and statistically hard to execute in particularly rare cancer populations. And so what the blood pack does, or the value the blood pack brings to the community, is if we can align in a pre-competitive space on, for example, the lexicon, what are the words we're going to use to describe our technology? What are the analytical performance studies and expectations, sensitivity, specificity, accuracy? And what are the clinical endpoints that we're going to seek and define? And what are we going to do kind of to frame these big questions into definable, tackleable, like achievable chunks, and then work together to kind of lay out that framework? That framework is extremely valuable for small companies entering the space, and also for advancing new technologies that we see great promise in, but are very difficult to design studies around when you're working in a vacuum. So that's the value of our community, I'd say.
Excellent. So
Lauren,
Peter, sorry, yeah,
Lauren.
Peter's got a question. So he's got to
No, because I mean, this is really important. What Jenn just described is really, really important for everybody to wrap their head around it, actually help us explain this better and quicker and in just a single sentence. Because and I love it, right? So when I listen to Jenn, and Jenn goes like, and then it's so exciting because we get to talk to the FDA, which Jenn truly enjoys. Okay. And when Jenn comes to my lab, right, Jenn looks at me, she's like, oh my God, this is awful what you do, Peter, right? And here I am, right, when I'm like, dude, I have no idea how this thing works, but I think I can make it work. Maybe once, maybe twice. That should be good enough, right? That is where I get excited, right? And then when Jenn goes like, let's go to the FDA, I'm like, oh God, no. That's like, do we really have to? I showed that it worked, right, once. That should be good enough. Let's get it out to patient benefit, because I'm a physicist. I know exactly what patient benefit is, right? So that is the complexity of this space. That's why so often, right, when we put a scientific manuscript out there, right, it's like, oh my God, we can detect cancer early. At this point, I have to say, well, detecting cancer early we solved like, I don't know, 5-8 years ago, roughly. The science of it. Exactly. Look at Jenn being like, dude, seriously? No, we haven't. That's the importance here, right? There's a huge difference between, hey, we can find cancer early, and making it meaningful. And the purpose of BloodPack really is to make it meaningful, because only then will actually the patient community benefit. And that transition is just, it's incredibly hard. And I wish that Lauren, Jenn, all of us, we could say, and in another 8 months from now, we will have all lost our jobs. That would be the ultimate dream, honestly. But that's just sort of the science perspective on the other side of it. Jeff.
Mute myself. I'm presuming the two people I know best in liquid biopsy, I'm assuming you know Mara Aspinall pretty well. And so she's written a great paper on the limitations. I think she published about a week ago. She writes a weekly newsletter on the limitations of multi-cancer early detection. And the other is, you probably know her equally well. She's in France, Alex Panabier. Kathy? So
yes, know them both.
Make sure that, because I know a lot of people are promoting the multi-cancer early detection, but the false positives are pretty limiting, as you probably know.
Yeah, absolutely. Absolutely. I would say, and that's why I kind of darted between terms, because I believe the field, and speaking to Peter's, like the research field gets very excited. Oh, we solved it. We solved it. It's there. There's DNA. There's there. It's going to work, right? But then when the rubber meets the road in a defined and controlled clinical study, what you realize is that multi-cancer early detection is actually quite a bit harder in large patient cohorts than you would think. And so where the field, I don't want to dismiss where the field has gotten, because where the field has gotten is incredibly impressive. What the field has been able to do is be able to define biomarkers that help you say in what organ or organ system a tumor may be appearing, and define kind of some of the signatures of cancer in the blood. Where the field is currently struggling, and you're absolutely right, is at first of all, defining and then achieving a level of clinical sensitivity and specificity that is meaningful for a large public health output. So when you look at the risk of a false positive, right, or a risk of a false negative, you have to look at the risk of both of those things, not just to the patient, because there could be a psychological concern from a false positive, and a false sense of security from a false negative, but also from a public health standpoint, reimbursement standpoint. What level of clinical sensitivity does an assay need to achieve to be able to be used broadly in the public, to be able to be embraced by the clinical community? And then what subsequent clinical procedures or screenings would you then? I don't think anyone in the multi-cancer early detection field would say, stop getting a mammogram, right? Like nobody would say that ever. But like crafting that understanding of where in the clinical space these tests are most appropriate, is it in the high risk population, the low risk population, and what kind of degree of clinical performance it needs to achieve to be kind of in the spotlight and ready for real time are active conversations in the field. You're absolutely right.
And let me add to that, Jeff, too. So what you mentioned there as well is, and again, Jenn just pointed this out, it's really, really important that we separate out the individual terms. When we talk about liquid biopsy, we talk about a much larger space.
No, I know.
Oftentimes, then we talk about the ctDNA tests. So, I mean, we have huge Guardant Health and others, right, have really delivered on actual specific tests for specific clinical utilities in managing patients with cancers. That is, so we really have to say that's amazing. The fact, and I somewhat flippantly said, that like 6, 7, 8 years ago, we solved the problem of early detection with a blood sample. Now, remember 10 and 12 years ago, if we would have had the conversation, I think we can detect cancer early in the blood, it would have been a ridiculous statement. It took years of just really, really careful, very high-end research to get us to a place where, hey, wait a minute, there is actually a signal of early cancer that we might actually be able to find in the blood. So the scientific breakthroughs have been absolutely outstanding. Coming to Mara Aspinall's commentary 10 days ago, whenever that was, really, really important. And in fact, it actually, in many ways, it summarized the blood pack session that Lauren and team just organized at the AACR meeting, and that had to do with what should, and this is what Jenn just pointed to, what should performance criteria look like? That is a really, really tricky one. And with that, we get to some of the other, with some of the comments that you made around false positives, for example, right? So there will, whether we talk about positive predictive value, negative predictive value, or false positives, false negatives, the multi-cancer early detection tests, remember they're called cancer detection tests.
Yeah.
They're not called screening tests. They're not called diagnostic tests, because those are specific utilities in the clinic, and we don't know yet. Right? We know that these tests can detect cancer. We do not know yet how to best use them. That is the work that's currently at hand. And that, again, has to be an equally thoughtful and careful process. I'll shut up now.
Let me jump in here
and Roger go and manage, and let Roger go and.
So why don't we go ahead and open up the questions? The way we typically do it is use the raise hand feature. And I see several people have had their hands up for a while. So Dinesh, why don't we start with you?
Yeah, sure. Hey, thanks. This is Dinesh from Royse. Very interesting discussion, and just wanted to sort of also add to that. So in terms of the liquid biopsy or ctDNA, it's not only about the MRD or early cancer detection. I think it could play a very important role in the comprehensive, tumor profiling also. And a lot of sort of new, papers are showing that when you use this, plasma as a sort of, complementary way, or the ctDNA approach as a complementary to the tumor, then you add a lot more variants than what you learn from tissue alone or tumor alone. In some cases, it could be actually you learn way more from ctDNA from plasma than the tissue itself, because tissue you might be taking from a particular site in terms of the metastatic setting or other settings, but in the plasma you may get more comprehensive picture. So if you combine both the tissue, tumor plus plasma or the ctDNA, then you can get a much more comprehensive picture. And in many cases, you could avoid then doing further surgeries, or you can decide on the what treatment type you could select and all that. So just wanted to add the another sort of application area, which is very, very interesting.
Okay, thanks, Dinesh. Richard has a question. Go ahead.
Yeah, I'm really sorry, I'll take these off. I'm in this space sort of as an observer, and it's really a complicated space in my experience because it is so easy to develop multi-omic tests with a lot of parameters and then convince yourself that the data is absolutely air-clad, and you've got great p-values, and you maybe even on the second study, since there are so many people trying, you get a great p-value the next time. And I guess when you do a drug, you kind of know what the mechanism is. You have a pretty strong Bayesian inference that it's likely these days to work, and then of course it doesn't work most of the time, but at least you have that kind of correlation. And in this, you just got a lot of different variables, and you're mixing them and matching them. So I've thought that it would be really useful to develop some kind of consumer guide to the studies, which would still make the problem really difficult, but at least have something like a consumer guide that helps patients evaluate this. And I know you're kind of an industry, partly an industry association, so you can't get into this, but are there certain tests that, well, I mean, I guess do certain tests reevaluate their clinical trial results in a phase 4 where they take a bank of what the patients are finding 2, 3, 4 years into the study? Are there any other things that people could use to make a determination of whether this is a great retrospective signal on retrospective data and maybe one lucky trial, but it actually is proving itself time and time again in the field?
Yeah, I'd say there's a fair bit of pressure in the field to keep going, right? Like you develop your assay, and to your point, I don't know how many times I go to scientific conferences and don't train to your test set, make sure you have independent assessment of your variables, make sure you have statistically sound methods going on. The blood pack has analytical validation protocols that we suggest people use, and we try to, and we do publish standards by which people should use to kind of establish the analytical and clinical validity of their test. But to the point, I would say right now, what's kind of putting the pressure on the field to keep going and give kind of real world evidence or continuing evidence of success are kind of reimbursement pressures in the United States, right? So to define clinical utility and identify patient populations that have benefited from these technologies, but also then in Europe, regulatory pressures to do post-market performance follow-up and other sorts of studies in the clinical population to make sure you're maintaining state of the art and the scientific state of the art. So I would say there is pressure on the field to move there. And I think a lot of very good providers are publishing a lot of very interesting papers as we start to get more clinical experience with some of these tools. But you're right, it's an ongoing saga.
So the regulatory pressure isn't coming on LDTs, laboratory developed tests for people's benefit. Those are tests where you can almost put the test out into the marketplace with kind of low levels of evidence. Those aren't getting those aren't getting a lot of regulatory pressure to do. post-market studies, but are there any kind of databases you could find of which tests, if you had a list of 10 tests you were considering, is there a database you could find that says easily, here's the tests that have been through post-market surveillance, here's what the testing looks like? Because that would really be helpful, I think, to people if they knew how to use it.
That's a great question. I see someone else has raised. Ari, was that something you wanted to try and answer that question? Because I'm happy to see.
Oh, no. I love the question, and I had a similar question, but I'll wait. I think that would be incredibly useful all around.
Yeah. No, I would say that's why I kind of highlighted kind of reimbursement pressures in the United States and then regulatory pressures in Europe. The regulatory status of diagnostic testing in Europe under the new regulatory paradigms of the IVDR don't have the same laboratory developed test structure that the United States currently has. And so it does get confusing maybe for patients about where you can get information about tests, though I would say that a lot of the major providers of liquid biopsy are prolific in their publications, right? So Guardant Health, Foundation Medicine, these sorts of companies do prolifically publish and are under pressure to publish, mainly because of reimbursement, to make sure they've defined the clinical utility of their test, and they are continuing to expand that clinical utility. But a consumer guide for liquid biopsy, I don't know, Lauren, like if we put that into our into our to-do list, but I don't know how we would make it quite yet.
Well, I would love to talk to you about how that might be done. I mean, I think it would be really important to be independent of industry, even though it would have to work with industry for obvious reasons. But if that were something that you think would be of interest, I'd love to talk to you about it. I think it would be really helpful to consumers and might sell a lot more product because consumers, would be involved in making sure this was useful to them.
Okay, Rick Davis has a question. Go ahead, Rick.
Yeah, hi. I'm not sure why my camera isn't working today. I'm sorry that can't see in person. So we at ANCAN have a long history in liquid biopsy and advocating, going back to probably, when I say long, going back to the mid-teens, which is relatively a long time ago. Remembering papers from the late Felix Peng and others on the equivalence of liquid biopsy to tissue biopsies and all of that. And I've worked with Foundation Medicine since about 2015, 2016. My question really comes back to Lauren, which is and this often happens with PACs, when I look at your PAC, I don't see the patient voice. And that really bothers us. The list of organizations that you have include ACS and PCF. The closest you've got to the patient voice is longevity, because that longevity does listen to the patients. But we at ANCAN it's all about the patients. That's what we're doing every day. And we feel that for advocacy efforts like yours, the patient voice has to be loud. There has to be somebody saying, well, this is how this type of test affects me and why I need it. Advanced cancer patients who don't know whether their disease has come back, talking about the importance of a ctDNA test. For example, in prostate cancer, where it's very hard for us to get ctDNA tests right now, we would say that your efforts are great. We endorse what you do, but where's the patient voice?
So I appreciate, and it's an interesting topic. This goes back, and I'm kind of looking at Peter. it goes back to the failure of our name, which is kind of, I don't think people want to spend time talking about this, but it is a failure of our name. We are not a political action committee. We are not at all. We are, it just happens to be our name. The Blood Profiling Atlas in Cancer happens to be our name. Many people do think it is a common problem. People think that we are a PAC, which we are not. We do partner with several patient advocacy groups. Longevity happens to be one of them. A few in the early detection space. Prostate Cancer Foundation, Breast Cancer Research Foundation, less patient advocacy groups than research groups, but they do have the patient voice. We have so far, our mission in the development and the approval of these tests has been very laser focused. So you are speaking to an organization that it's not that we don't seek out the patient voice and do engagements like this, where we are trying to receive more feedback and content from patients and be able to reflect back. But the majority of our work, almost all of it, in fact, even our education project that will focus on accessibility, essentially for patients, is actually focusing on the education of clinicians themselves. So what role do we, as primarily diagnostic developers and pharmaceutical companies, based on our expertise in the field, what can we contribute to helping educate physicians specifically? So we do not attempt to say we should be educating patients or patient advocates at all. We are laser focused, and we appreciate that that is a role that needs to be filled, but there are really spectacular organizations that are doing that and doing it very well right now. And we have taken the approach that we would like to maintain our focus on the very relatively narrow space that we fill, creating these standards, creating these frameworks for the industry, and collecting the data to support them, and then provide that content to other organizations who are able to translate that content and have the expertise to translate that content to patients. That said, again, we greatly appreciate the patient voice and have a few of those select organizations like Longevity or Prevent Cancer who sit on our accessibility kind of overarching working group, who review the content that we put out there. So if we create a lexicon of terms we have a dictionary online that I encourage everyone to go to. I think it hopefully will be helpful to patients for around MRD, around early detection. It'll eventually be around CHIP, that content has been reviewed by a few patient advocacy groups in an effort to say, is this going to be helpful or meaningful to patients? Are they going to be able to understand it? However, we have found that the majority of our lexicon and definitions will never reach the patient. So they are very extremely scientifically based. It's required for them to be kind of stringently scientific, and most patients will never hear those terms. And so we are kind of a layer above where patients actually hear us. And so it's important to remember, it is not that we don't. Does that make sense at all?
No, not really, to be honest. The reason it doesn't make sense, and I've got some of our advisory board members are actually on this call, and I think they would sort of back this up, that it's a very patronizing approach to the patients that you're taking. Yes, you need to address the physicians, but half the time, people that we navigate finish up addressing the physicians and teaching them what they need to know because they're hearing it from us. So I think it has to be a collaborative effort that includes both patients and physicians in developing your message. I also feel that when you talk about PCF or some of these other organizations, they're not the patient voice. They have people in there who speak for the patient, but do they send out your terms to the patients and ask the patients, can you understand them? They don't. And if you drill down on them, why don't they? Because they don't have the contact with the patients. They may represent the patients, but they don't have the contact. So what I'm saying is if you have somebody like a Lenciera on that on your board. You're actually hearing it from the patient, from a person who has a deep scientific understanding, who is a peer, who is a patient. And these are the sorts of people you need their input as part of what you're developing, and it isn't represented. I would ask you, and happy to help you, happy to work with you, happy to point to you. You were just at AACR. Ben Nathanson, one of our guys, had a poster in there and on ANCAN and what we're doing, and he's deeply scientific. We have these people who see it from both sides. And I think organizations like yours, forgive me for mistaking you for a PAC, because I clearly see you're not, but I still think in developing your message, you need to hear it directly from the patients who may be educated, but they're going to help you how to explain your message to the patients who are not educated because they see both sides.
Okay, thanks, Rick. I think it'd be a good thing for you guys to follow up with.
I think
maybe
Peter, though,
afterwards.
Peter, did you want to say something too?
Yeah,
But
just
I think other people
again,
like-
because
Sorry,
the nuances on the words that we're using are really important here. And I just want to make sure that, and Lauren pointed this out, right? We are not an industry organization. So when Lauren just used the word in the industry, she actually meant the entire space of liquid biopsy, which is participated in by academic groups, just as it is by industry groups. In fact, within BloodPAC, while the majority of members are industry folks, right? It's their scientists that are part of the group. In fact, we are more scientific driven or more academically driven than most academic organizations are. So it's really important that because somebody else asked about on-market validation studies, etc., and whether or not we can talk about it. Again, it's really important. BloodPAC is not an industry organization. It doesn't represent industry. It's really meant to support the process of development, validation, and accessibility of liquid biopsy, very much in this context of, and the reason why I played this bit back and forth between Jenn and myself, right, is so that Jenn and myself, so the people who actually are part of this process from the very beginning all the way through regulatory approval, are actually aligned of what's required. Because typically people in my spot don't actually understand what it takes to get to that finish line. And doing this in a public-private or in this public setting of BloodPAC as a nonprofit really educates everybody across the board. And that is really the purpose of this entire organization. And I just want to make sure that that's actually clarified.
Okay. Thanks very much, Peter. Let's move on. Alan Morris, you have a question? Sorry, Alan, we can't hear you.
You're muted.
Alan?
Hi, I have a lot of questions. I'm going to start with a question I think I already know the answer to, which is I'm going to go through the individual spaces and ask if you have any recommendations. I think Richard already asked you, and I thought the hint was that since you have a lot of like you have Natera and Garden and Tempest and Foundation and all these people that are in your consortium that you probably can't recommend. But let me ask you, because I really didn't hear it. Let's take multi-cancer early detection. Do you have a company you recommend?
So I don't recommend any companies over one or another. I would only say that I am strongly, suggest that you take time to look at the companies that participate in BloodPAC who are willing to share their information, their pre-competitive content and data to improve tests for the community as a whole. I tend to be relatively skeptical of the companies at this point who some are too small to join BloodPAC, so that's another issue. But there's some that are, I don't want to work in BloodPAC because I don't want to share my learnings and to help benefit the industry as a whole. And to that I say, if you come across companies that are larger who aren't on our list of companies, I'd probably ask, have you participated? But we have a good number we have a working group, early detection and screening working group. So those who are working in single cancer screening as well as multi-cancer early detection. I don't recommend any one over another. I actually know Grail just came out with some positive data today. I can put the link into the chat if you'd like me to. But they all work very well together. I think it didn't used to be the case several years ago. They're extremely collaborative now and host a lot of discussions, but I'm not qualified. Everyone should know I'm neither an MD nor a PhD. I'm a lowly MBA. So I am not qualified to make that assessment, even if I loved one over another.
Okay, in the diagnosis space, it's not just early detection. I can tell you the first space of that as a pathologist would be there's an entity that used to be a very common entity called metastatic cancer of unknown primary. The reason I'm listing this is I would ask you for a recommendation for that part of the diagnosis space, but I have experience, and the experience is, for whatever it's worth, Foundation 1 is used most frequently by our clinicians when we have a case of metastatic carcinoma of unknown primary, where they're trying to decide what's the primary site so they can go into for example, NCCN guidelines and decide what the best treatment is for a patient. And I can tell you that Foundation 1 can be and is not uncommonly wrong. And that just speaks to the thing that people don't look at that are lay people, which is false positives and false negatives. You mentioned Grail. Roger is our case in point. Roger would testify for Grail because he unbelievably has a story where he had a Grail positive signal. They did an MRI and detected his early pancreatic cancer, and he appears to be no evidence of disease at this point. So he's a miracle of Grail, and he could get up and be a Grail salesman. But Grail, I'm sure, suffers even more than Foundation 1 in the concept of false negative and false positive, and why it is still not FDA approved. Now I'll go on. I want to make a comment because I want to tell you what real world experience is for a pathologist. Now I'm going to go on to minimal residual disease. It turns out the most penetrated company in our community, part of it is commercialization and marketing, is Natera, followed by Guardant. And Natera has the most advanced studies in colorectal cancer. And in fact, our clinicians are ordering Natera for minimal residual disease monitoring in people that are for people that have colorectal cancer, breast cancer, urothelial cancer. And there's a debate in our community, and most of our oncologists will say that there is no evidence yet for any of them for improvement in overall survival. And so what you are literally doing that people would agree is getting lead time bias. You're getting a lead time, maybe it's 6 months earlier or whatever. But for example, colorectal patients are faced with things like Fulverox, a pretty strong chemotherapy regimen. And our clinicians, our oncologists in general aren't going to be I guess I'm taking a step backwards, and the push here is patient advocacy. Geez, we've got to educate our clinicians. Our clinicians are educated. They're not dummies. They're hearing this as much as all of you people that are patient advocates. They just are waiting for efficacy signals. Because, and unfortunately, it's a bad word here. It's called standard of care and what's approved and all that kind of stuff. But basically, you're weighing efficacy versus the risk of all these, treatments that have high. I'm starting to go into a big.
Ellen, let me stop you there, and let's let Lauren and Jenn respond to that.
So I'll ask the question. Do you recommend a minimal residual disease company, and for what cancers do you recommend them?
Okay, so I'll one thing I will say with respect to comprehensive genomic profiling and real world evidence, your experience echoes our own. Like, we put out comprehensive genomic profiling, assuming our majority patient population would be non-small cell lung cancer, because that's the cancer population that has the most kind of precision targeted therapies. And we found more than a third of our volumes were actually, if not properly unknown primary, it was where the pathologist was between two, right? Or it's like, it could be either this, or it could be maybe this, or it's either this primary or a reoccurrence of this, like previous malignancy, these sorts of things. And so that has actually been, yeah, that echoes our own experience, which is comprehensive profiling we thought would be a very straightforward, like, it's going to be lung cancer patients looking for their precision therapy. And in the end, I'd say most of the utility comes from like that kind of refinement of diagnosis that I think has been valuable in the field.
Do you recommend a company for metastatic carcinoma of unknown primary?
There is no company that I know that actually makes an explicit claim about metastasis of unknown primary. Even Foundation, I don't believe, makes an explicit
No, they have a
claim
test.
to that.
They have a test called Foundation One, and it is marketed as such, at least to us.
Yeah. All right.
Okay.
I'm looking at Rick, so I won't address, Jen is our guru, what tests are out there, but I'm looking at trying to follow all the comments and discussion. Rick, I actually agree with you. We have found that you are a extraordinary group of individuals and patients who are intentionally driven and knowledgeable about, in many cases, your own disease or others' diseases. And as well as clinicians who work perhaps at Memorial Sloan Kettering or MD Anderson, who are extremely knowledgeable about these tests. The reality is, I think someone just told me, and Sandra, I'm actually looking at you, you probably have this number better. Like, there's like the 80-20 rule. 20% of patients or clinicians know really about these tests, and 80% just don't know. And that made me generous too, I think. And so I tend to agree with, Alan, I have to unfortunately disagree with you. Like, I just don't think most people are aware of these tests or knowledgeable in any real meaningful capacity.
Well, certainly not primary care physicians. And Ellen mentioned my name, so I'll tell you, I'm Exhibit A. Now, I went to, I've corresponded with the U.S. Preventive Task Force Service, which recommends against pancreatic cancer screening on what Ellen said. You're just getting lead time, doesn't change the outcome. Well, I'm Exhibit A for the fact that that's wrong. And I not only got a liquid biopsy, it wasn't without the advice of a physician, it was against the advice of my physician. He said, don't do it. They don't work. You're just going to get a false positive and it's going to scare you. And one of the very few things that my doctors agree on now is I probably wouldn't be here if I hadn't done that. So you can make all the statistical arguments you want. I'm with Rick, on patient advocacy. I'm not a statistic. I'm a person, right? And I wouldn't be here without it, probably. We got to move on. Ari, a lot of questions.
Can I just add one thing on this? And I'd like to point Alan to the research that's been done by Mani Tieno at Illumina and Neil Shore on testing frequency. What we're seeing is exactly what Lauren is saying, that the take-up by community oncologists of genomic profile testing is just atrocious. I mean, if you look at their actual research and show how many people tested for the existence of a PARP sensitive mutation, it's really low. They had a poster this year at GU ASCO. They had a poster last year at GU ASCO. So we know it's really poor.
Okay.
Okay, since no one's going to call on me, I'm just going to unmute and jump in. Okay. Sorry, I got here late.
Sorry, Ari, you've been jumped over. Go ahead, Paul.
Because it's pertinent to previous discussions. about activist patients who are very involved in their care and do research their options, like the people who are here on this forum and other forums, and myself included in the advanced prostate cancer space. And we take the information to our physicians and we ask for things. We promote the use of technologies of benefit for us directly to our physicians, as I did recently, because I'm interested in MRD detection in my own body from my treated and apparently now indolent prostate cancer. So using circulating tumor DNA detection to see if there's something growing that is not being picked up by my PSA. My oncologist said he didn't know anything about this. And I said, well, I want this. Could you please research it and find out how to get it for me? Should I get circulating tumor cell counts from, or should I get a ctDNA screen, quantitative, to what percentage of my circulating DNA is tumor DNA? And then if it's detected, can it be genomically profiled? And I said, I want you to do this. The precedent for this is in the pharmaceutical industry. Turn on your television and see how many very expensive new drugs are being promoted directly to patients to go ask your physician about them, because that's found to be a channel that works. So I disagree with ignoring the patient orientation towards promoting wider appropriate use of liquid biopsies, not in just my cancer, but in many cancers.
Okay, thanks, Paul. All right, you had your, okay, I guess you've withdrawn your question.
It's
So Sandra,
fine. Alan basically got to it. I was going to ask, following up on some of the earlier threads Richard had asked, and Alan, who isn't getting liquid biopsies, just, given the state of play today? Are there one or two, not multi-omics, but kind of single? I heard maybe it was lung cancer, non-small cell lung cancer was maybe a good use case, but who isn't getting this that should be getting it today? However specific, however pointed the test actually is. Yeah, I'd just be curious, very simple.
So I think I will probably weigh in and start with maybe even the base case of tissue assessment for comprehensive genomic profiling. And I'd say there's been several recent publications that indicated that it was approaching 75% of advanced non-small cell lung cancer patients are getting kind of comprehensive genomic profiling. And then the numbers fall precipitously by tumor type after that. And in the words of the founder of Personal Genome Diagnostics, I view it as a sin, right? Like in an advanced cancer space, to not have comprehensive genomic profiling of some sort in the era of precision medicine with so many targeted therapies is, I think, a real and present danger to our advancement in this space. And I think the hope of BloodPAC is that for patients who can't get a tissue-based or a tissue-based comprehensive profile, like a liquid biopsy-based would minimally be executed upon. But I would say recent data says most patients are not getting liquid biopsy, right? And even those that maybe would most benefit from it, particularly in non-small cell lung cancer and other tumor types that have a lot of precision targeted drugs. So I'd say that advocacy, that's why Lauren and our group has added an accessibility arm to our organization, is to maybe address that kind of problem in our field, I'd say.
Okay, thanks. That's too bad. Sandra, you had your hand up for a while. Do you still have a question?
Yes, thank you. Well, thank you, Lauren and Jen, for being here today. My question is about, you are in the front line working with all the organizations. When do you think that minimal residual disease or molecular residual disease is going to be standard of care, time-wise?
So I think it's been discussed maybe already, but it's going to depend on tumor modality. And I would say, of course, minimal residual disease is assessed in hematological malignancies today as standard of care very normally, right? And I would say probably in the case of colon cancer is the solid tumor modality that probably has the most clinical evidence to date. Following behind that, probably lung cancer, bladder cancer, et cetera. But I do think this is going to be a kind of indication specific, tumor modality specific, and kind of stage specific rollout of the use of molecular residual disease. Right now, it's like trying to identify high risk patients with maybe stage 2 colon cancer who you're trying to decide between like surgical intervention could be curative versus needing a round of FOLFOX, as has already been kind of discussed. And so that seems like maybe the first most obvious hard clinical utility evidence that's moving into the field. I'd welcome anyone else's thoughts, but I'd say it's going to come slowly, similar to how minimal residual disease has kind of integrated itself into the hematologic malignancy space. It'll come into solid tumors, and it'll come indication by indication. Yeah.
Okay.
Thank you, Jen. And if I can, Roger, just one more question.
Okay, last question. Good.
Is there anything that an organization, a patient organization, such a cancer patient lab could do in order to help to accelerate the approvals or implementation of any sort?
I'm not sure if I have a lot of good advice on that one. With respect to anything, Lauren, do you have any points to consider?
I don't, I mean, I don't want to speak. And again, this is why I wish Rick was still on. I would like Rick to answer this question as a better person to answer. You know, again, from my perspective, I think our success has been driven by us staying, and Sandra, you're welcome to join since you are a member of BloodPAC Roche's. You're welcome to join any of our meetings, but essentially staying in our swim lane of, hey, we are helping to develop these tests collectively. I wish I had also said when Rick was on the phone, we have invited many patient advocates individually and as parts of other groups. And we do have several colon cancer groups now as part of BloodPAC to join our conversations. They find it extremely difficult to keep up with or be able to do exactly what you're saying. It's not keep up, translate the science, the minutia of the science and the details of what we're trying to create in the development of these tests to something that's valuable for even a patient or a clinician, which is why there is a whole separate working group that we have to say, how now we have a test, how do we get you to actually use it so that it can ultimately, you know, be approved and you know, used in standard of care. Although there was an argument yesterday on one of our calls about what is standard of care and how does one define standard of care.
Correct.
Right. So, I mean, again, we are so far upstream from a lot of these conversations. I wish there are so many things for groups like this to be doing. I just don't, I struggle with trying to identify how to best incorporate specifically into BloodPAC, unless it's indication by indication or disease by disease. We do have, and I'll say, Roger, I know you're just wanting to get us out of here, but, you know, we do have a brain tumor working group, specific brain tumor working group as well, that we don't have any other disease specific groups. And in that group, we do have funding brain tumor foundations that are funding in the brain tumor space. We do have one patient advocacy group. We have several academics. So the majority of our academics actually sit in that group because the brain tumor industry within industry is so small so far. We have to lean heavily on our academics. And so there is feel like an interesting space for our advocates in that area specific to brain tumors. But other than that, I do struggle to identify where within BloodPAC we can best utilize, other than also having on our sessions at like AACR, we have had Prevent Cancer and others as the voice of the patient sit on our panels and say why the creation and continued creation investment in the multi-cancer early detection space is relevant and useful to the community as a whole.
I agree. And thank you, Lauren. Perhaps, as you said, down the road, once the test evidence is ready for guideline inclusion, perhaps that would be a good point for some interaction. Thank you.
Okay, with that, we're over time, but we've had lots of questions. I don't see any more any more hands raised, and we'll put in the show notes the contact information for Lauren and Jennifer, because I know that there are probably people that want to have further discussions and especially want to check out your organization. I want to thank you for being here and thank you for the lively discussion. That was a lot of questions and a lot of audience participation from one of our groups. Okay, it's Roger Royse, Cancer Patient Lab, and we've been talking with Jennifer and Lauren about latest developments in liquid biopsies. We'll see you all next week.